This is a demo of pVACview. I will be walking through the steps of launching pVACview, either through Terminal or RStudio. Afterwards, we will walk through the steps of uploading, visualizing, exploring, and eventually exporting your results, all using a demo dataset. Starting off, we first need to launch the pVACview app. This can be done in two ways, either through Terminal or RStudio. I'll first demonstrate this process using the terminal. In Terminal, we can first check the necessary inputs to the pVACview command. As you can see, in order to run pVACview, you simply need to supply your pVACseq results directory. There is also an option to specify the location of R to be used for launching the app. This is convenient in cases where you have multiple versions of R installed. Now let's launch the app using the pVACview run command and supplying the location of our demo dataset as input. It might take a couple of seconds to launch. And once successfully launched, your R console will output a host address that the application is listening on. Simply copy-paste that into a browser of your choice. Here I am using Google Chrome. And hit enter. This will take you to the upload page of pVACview. Next, let's try launching the app through RStudio. First, navigate and set your working directory to where your pVACview R files are located. Open either the app.r file, or the server.r, or the UI.r file. All three, once opened in RStudio, will have a Run App button appear on the top right corner. Simply hit the button and this will launch the app in the RStudio browser. However, we strongly recommend to copy the host address located at the top left of the browser window and launching that in an external browser, such as Google Chrome, for its full functionality. Once the app is launched, we can either load in a full demo dataset from the HCC 1395 cancer cell line, or we can choose to upload our own dataset. If you have any questions regarding usage of the app, we have a help button on the top right corner, as well as basic instructions with links to individual steps in our pVACview documentation. Here, I will walk through the steps of uploading all the required and optional inputs using the HCC 1395 dataset. There are two required inputs for the app. First one being a TSV file containing an aggregate report of all neoantigen candidates. Users will then need to specify whether it is a Class 1 or Class 2 data being uploaded. The second required input is a JSON file containing all metrics data ranging from the variant to transcript to peptide level for all neoantigen candidates. We also offer the option to upload additional report files with a label specified. This is convenient for users who, when analyzing Class 1 data in detail, also want a basic idea of Class 2 results for their candidates and vice versa. Last but not least, users have the option to upload a gene of interest list in a TSV format with each gene on an individual line. These genes will be highlighted in the main aggregate report table in the Explore tab. Once you have successfully uploaded your files, go ahead and hit Visualize. This may take a couple seconds, but the pVACview app should direct you to the Explore tab shortly. The first thing you see upon switching to the Explore tab is the main aggregate report table. By hovering over each individual column header, you will be able to see a short description for the respective column. For columns such as the predicted IC50 binding for mutant peptides, we have overlaid a coloring scheme with different shades of green representing binding under 500 nanomolars, and shades of yellow and orange representing binding between 500 and 1,000 nanomolars, and red representing those above 1,000. Similarly, the cutoffs for the percentile column are 0.5 and 2%. All the candidates are given a tiering based on if they pass certain criteria. The specifics of this tiering metric is documented in our help docs. For each candidate, we use red boxes to highlight which criteria the candidate is failing. Candidates are automatically sorted with passing candidates in the beginning of the table and candidates with no expression at the end. Now let's pick out a candidate to investigate further. Clicking the respective Investigate button located at the end of each row will allow the app to search and load transcript and peptide level data for the variant of interest. Scrolling down, we can see more information presented in the box below. On the left, we have the transcript sets covering this variant that produce strong binding peptide candidates. These transcripts are grouped such that if multiple produce the same set of peptides, they then belong to the same set. Here we have two transcript sets, one containing 8 transcripts and the other with just 1. The first set produces 6 peptides, while the second set produces 10. Total expression is calculated by adding the transcript expression values in each set. If you choose to upload additional data, the second tab on the left will show the median IC50 prediction regarding the peptides containing this variant. In this case, I uploaded additional class 2 data. The middle section contains some additional variant and gene information, such as DNA, RNA-VAC, gene expression, and genomic information. We also provide a link out to the corresponding Open Cravat variant report. This report contains additional information, such as clinical relevance, functional studies, and related literature, if any is found. Moving down the page, depending on which transcript set you select, you can then look at the individual peptides generated from that transcript set. We also provide a second tab that showcases the specific transcript in each set, with individual expression values, TSL, file type, and transcript length. The peptide table contains the amino acid sequence, as well as the median predicted binding for the different HLA alleles if the mutant peptide binding was under 1,000 nanomolars. By clicking on each peptide pair, the plots below will change reactively. We have 4 different tabs containing additional information on the peptide level. There are 2 score plots, one in nanomolars and the other in percentile, both showing the distribution of individual algorithm prediction scores for the mutant wild-type peptide pair. This can help give the user an idea of how the algorithms agree or disagree on the predictions. We also have a score table that listed the exact numerical values of the algorithmic predictions across the HLA alleles. The last tab showcases the peptide sequences with a heat map overlaid. This heat map shows the probabilities of each position acting as an anchor location with a mutation marked in red. For users needing guidance on how to use this anchor information, we also have a cartoon scenario guide on the right. Once the user is happy with their analysis of the variant candidate, they can label it by changing the status of the peptide to accept, reject, or needing further review, such as genomic review in IGV. All evaluations are updated in the peptide evaluation overview box below to help users keep count. Users may also add additional comments for the variant that they are investigating. As an example, the variant we were just looking at has multiple strong binding peptides across multiple HLA alleles and has good expression on the transcript level as well. No major concerns, and would recommend for things such as downstream vaccine design. These comments are then saved and will show up in exported files if you choose to do so. Last but not least, we have some advanced options to allow the users to update the tiering of each candidate to their specific need. The first thing that users are able to change are the anchor locations of the peptide MHC complexes. By default, anchor locations are calculated as 1, 2, N-1, and N for any M-mer peptide. Users can choose to utilize a set of computationally predicted allele-specific anchor locations to replace the default setting. For more details, please refer to the pVACview documentation and the linked manuscript. The second thing users may want to adjust is the VAF cutoff for determining clonal versus subclonal variants. By default, this value is set to half of the maximum DNA VAF under 0.6 across the entire sample. However, users often want to adjust this cutoff according to things such as the VAF of driver mutations observed. Lastly, allele expression cutoffs are used to determine tiers such as passing and relaxed variants. It is also one of the criteria for subclonal and anchor variants. Hence, we also provide users with the option of changing the default values according to their specific sample. The details of how each tiering is labeled is also documented at pvactools.org. Once the users are happy with their evaluation and annotations, they can move on to the Export tab. First, users should change the export file name as needed, and then double-check the comments and evaluations that you have made. We offer two file types for download, TSV and Excel. If the user has not finished their neoantigen analysis, simply download the TSV file to store the comments and annotations, and you may reload the downloaded TSV file as your main aggregate report file upon re-entering the application at a later time. Note that pVACview does not save any of your data once it reloads. Hence, saving through this method is critical for maintaining your evaluations. That is the end of our demo. If you have any further questions or feature requests, please contact us through the pVACtools GitHub repo.